All right, so explanation of the glucose use diagram. We'll go right down here to a cell, okay? This cell has mitochondria in it, and it's doing cellular respiration. So we'll get to that in a minute. Surrounding the cell is smallest blood vessels in the body. They're called capillaries, and these capillaries are actually made up of one layer of very, very thin cells called squamous cells, okay? And in that capillary, there are a couple things, one of which is oxygen and glucose. All right, how did the oxygen and glucose get there? I'm glad you asked. Well, the oxygen gets into the bloodstream through our lungs. So we inhale room air, which contains oxygen, and that that oxygen will then come in through these small airways, past the smooth muscle, look like loose rubber bands, and end up in these air sacs. They're called alveoli. And these air sacs are also made up of individual cells that are simple squamous, one layer of very, very thin flat cells. So we've got a high concentration of oxygen in the air sac. We have a low concentration in the blood vessels that are surrounding those air sacs. So because oxygen is small and uncharged, it's going to easily diffuse through the phospholipid bilayers and go from a high concentration to a low concentration down its concentration gradient into the bloodstream. And then in the bloodstream, it's going to get carried down here to the cell, okay? And same deal. There's a high concentration of oxygen in the capillary, low concentration in the cell, and it's going to go from high concentration to low concentration down its concentration gradient and diffuse through simple diffusion through the phospholipid bilayers without any problems whatsoever. Okay, so that takes care of oxygen. Glucose. How do we get glucose? Well, typically we'll eat complex carbs. Okay, and those complex carbs will get swallowed down your esophagus, which is your food tube, end up in your stomach. So we'll use pasta here as an example of our complex carb. Okay. So after staying in your stomach for a little while, the pasta is broken down mechanically, not enzymatically though in the stomach, but because there's no enzymatic catabolism or breakdown of carbohydrates in the stomach. It's mostly proteins. So anyhow, that pasta, those complex carbs are going to end up in your small intestine, and after spending some time in your small intestine, those complex carbs are going to get hydrolyzed or broken down into simple carbs. So here we have glucose in the intestine, okay, in the small intestine. The glucose is going to go from your intestines into the bloodstream because your intestines are wrapped with capillaries, okay? So this is how we get glucose and oxygen into the bloodstream. Now, we need to get the glucose into the cell in order to do cellular respiration. The problem is that glucose cannot enter the cell easily. It needs a hormone called insulin, which allows it to enter into the cell. Okay, so we have an asterisk there, and our little key is up here, insulin. All right, so if we have insulin, we can bring glucose into the cell. Think of glucose as a $10 bill. So the first step is extracting. In order to extract all the energy we can out of glucose, we're going to get a lot of ATP. It's like 36 ATPs. Okay, so think of ATP as a quarter. Okay, so if I have a $10 bill worth of glucose, that's 40 quarters worth. So if we look at the end of the process here, if we look at the mitochondria as basically a change machine, the mitochondria is going to produce CO2 and water, we'll get that in a sec, and 36 ATP, 36 quarters. That's pretty decent. If I put a $10 bill into the change machine and I get 36 quarters instead of 40, it's pretty good. I can live with that. Okay. But the first step is, we need to do this step by step by step by step. We can't just go from a $10 bill to 36 quarters. Okay. So we need to split that glucose molecule, C6H12O6, into two pyruvates or pyruvic acids, and we'll consider those $5 bills. That happens through glycolysis. With glycolysis, it's hydrolysis of glucose. So you're adding water to glucose and splitting into these 2 3-carbon molecules. Okay, I'm skipping a lot of steps here. Okay, so the pyruvates are going to enter into the mitochondria along with the oxygen, and a whole series of chemical reactions, Krebs cycle, electron transport chain, are going to occur in here, and the products are going to be CO2, water, and 36 ATP. The ATP is the reason that we do cellular respiration. We need ATP to use as energy. Okay, yes, glucose is energy, but it's not usable energy. Just like the $10 bill is not usable if you need to make change, and you can't make change. It's not usable. So anyhow, there's a buildup of CO2 within the cell. CO2 is going to diffuse easily out of the cell because it's only 3 atoms and it's small and uncharged. It's going to go from high concentration to low concentration in the bloodstream, and we're going to get rid of this CO2 in the lungs some other time. And the water is also going to build up in the cell and diffuse through osmosis into the bloodstream. Okay, now that is great when you have oxygen. Okay, there's another thing we can do too, because if we don't need this ATP right away, it's kind of silly to go through all this and have all this ATP sitting around. So you might as well have that glucose. If we have extra glucose around, might as well have that glucose come into the cell again with insulin. If you don't have insulin, you can't do this and you can't do that. So we need insulin to bring glucose into the cell. And now we're going to polymerize this through dehydration synthesis. We're going to take all these glucose molecules, and here's our key right here, triangle. We're going to rip an H off one and an OH off another, so we're going to remove water, dehydration, and put these guys together, synthesis. And the end result of that is going to be a molecule. The storage molecule for carbohydrates in animals is called glycogen. We store lots of glycogen in the liver and lots of glycogen in our skeletal muscles because our skeletal muscles need a lot of energy. And when we run low on ATP, it's easy enough to tap into our glycogen stores. Now, in order to tap into the glycogen stores, there are a couple different ways we can do it, okay? If glycogen is stored in the muscles, skeletal muscles, which it is, adrenaline can start the process of breaking apart glycogen, okay? We also have a hormone called glucagon, and glucagon works not on muscle cells, but it works on liver cells, which is why the liver stores a lot of glycogen. So let's say that we're running low on glucose in the bloodstream because your body does not care about how much glucose is in the cell. All your body cares about is serum glucose, how much glucose is in the bloodstream, and there's a certain set point that we'll get into later on. Okay, so let's say you haven't had breakfast yet and we're running low on glucose. Okay, not a big deal because we can go over to the liver where we have glycogen stored, and we can, using the hormone glucagon, we can break up glycogen through hydrolysis. So it's the opposite of dehydration synthesis. Hydrolysis is using water to split apart glycogen into individual glucose molecules with an OH hanging off them. Okay, and then that glucose can make its way out into the bloodstream to raise our blood sugar back to normal. Okay, that's only if we have oxygen present. If we don't have oxygen present, we run into a little bit of trouble that we'll learn about later on in this course. So again, the key concepts are oxygen easily diffuses in and out of cell membranes. Glucose, however, needs the hormone insulin to get from the bloodstream into the cells, and we need the hormone glucagon to catabolize glycogen into glucose from the liver cells into the bloodstream.